Today I would like to go with you a bit through some bit of theory and then some examples of ways we can visualize and especially since we want it to be interactive, so explore our spatial transcriptomics data, because since this goes beyond images and sequencing, but we have tables and images and points and shapes. It's quite a complex data set and requires a bit more also complex setup for really being able to make the most out of our experiments. So our webinar, first of all, I would like to do a short recap or maybe a short introduction on the spatial data format. This is what I'm going to be using with all our examples for managing all the spatial transcriptomics or spatial proteomics data. I'm going to briefly discuss how we can store, host, and then share the data for the visualization. And we'll go through a bit of a summary of some series of nice open source visualization tool. And then we're going to work together to a set of examples. So now we can start it. And okay. So thing is, managing. Why am I starting from the managing of spatial omics data? This is because, well, it's not exactly trivial. Currently, datasets are tens, hundreds of gigabytes, now slowly moving more towards the terabyte range. So it really quickly gets very cumbersome having to move them around, either to local hard drives and then going from the instrument to the local PC in the office, maybe upload them to central server, and then waiting for copying, upload, and download. And this is already a lot if one is doing the analyzing its own data, but if one wants to then have a more cooperative workflow, cooperative approach with multiple bioinformaticians, or maybe then share the visualization with an histologist or a pathologist, to get some feedback on their annotations. Maybe you have an H&E attached to your slide image attached to your data, and you can want your pathologist to add some annotation on top, or you want to have an expert check, do these cells really look like the cell types that I'm calling? Then it becomes really useful to being able to share the data effectively. And maybe it's not really necessary to share the whole data set. Maybe we could just give them a link to the data set up in a way that they can directly explore and view from the browser. And this is not the only reason. It's one of the reasons why we like to go and work with the spatial data format. And this has, let's say, multiple reasons. First one is that it tries to solve a huge fragmentation problem with many different vendors having many different formats that then need to be, well, just how am I going to open the images and view them? This is already quite tough, especially if you might have seen the same aspect in the microscopy world. Now, why SpatialData format instead provides a lot of convenient pre-made readers that can take the output directly from their different instruments and convert it into a unified framework. But it's not only the use of a unified framework, it's also the fact that it's based on the OME NGFF standard, meaning that it can handle images, points, shape, labels, and tables all together, all in the same container. It works really nicely with streaming or web download, or with object storage. So like if you have access to S3 buckets, and it becomes really efficient for working and sharing with this. And SpatialData object works as a container for various elements, and this cover all the type of data that are produced in the typical spatial transcriptomics experiment. For instance, you have contain images, meaning everything from your H&E to staining to multiplex immunofluorescence to just overview of the slides. Then you can have your segmentation at the pixel level mask, meaning cell nucleus segmentation, or also tissue annotation, meaning this part of the image contains tissue, or this is tumor, this is stroma, etc. You can have, or you can represent this region of interest as shapes, so the boundaries of your cell nucleus, or again, anatomical notations. You can have points, meaning a dimensionless set of coordinates, and these will generally be used a lot to represent the location of transcripts, and we're going to see it in some of our examples with Xenium data, for instance. There, the transcript location is generally just represented at point. And then finally, either directly from the vendor, in case on the instrument, in case of the Xenium, or after a bit of analysis, you also get tables, which are matrices that generally annotate the other elements, meaning the shapes and the labels. And these are tables which then contain your cell type annotation, your PCA and UMAP coordinates, of course, the expression matrix with the transcript or the protein expression, or even both at once. And these tables are in the, let's say, background. They are undata objects with which some of you might already be familiar from the single cell analysis world and the whole SCAMPI ecosystem. And another thing that I'd like to point out here at the bottom is that it's not just a container for all the assignments, it's also a very nice way to tie everything together, meaning that the annotations in the table are linked exactly to the labels and shapes they are annotated. But also, if you start having more complex workflow, experimental workflow, where you are going maybe for some multimodal experiment, meaning you run your Xenium spatial transcriptomics and then perform Comet on it, so the spatial proteomics experiment on top. Then you can align the data sets through different registration workflow, and then the SpatialData object also helps you taking care of the transformations that align together the data from the different modalities. And if you have time, maybe you also see discovered in one of our examples. And finally, I introduced a little bit before, but other big advantage of spatial data and the underlying data for the table is that it's based on the SAM format and on the OMI and GFF on the image size, which means that it supports parallel read and write and allows you to access the single components that you need. You don't need to load 20 channels in 3D if you just want to quickly check how your DAP is staining look. And you also don't need to download the whole data set, which could be very easily a couple hundred gigabytes just to do a very quick exploration. Negative side, this is still, again, very pretty new format. So the support in terms of tool is not as much as we're used to working with TIFF files or the sequencing side format. But I would say this is already catching up very, very fast. Okay, and then we get, of course, back to the problem. Okay, now we have this beautiful format to keep our data in, but where do we keep it? So they could be either in cloud or object storage, and this is probably the most efficient way. It's the way that allows you to share the data and use them with web viewers like Vitessce in the fastest, most efficient way. Cons is that in most cases, most tools still need full public access to the bucket, which might not be, which is not often what we want for our data, at least not before publication. And very similar side on the public repositories, which are now beginning to offer more and more also directly themselves this kind of visualization. Here I have a quick example from the UpMap consortium, but again, we'll catch up with more on this side at the end. And of course, we can keep them locally, either local machine, but of course, space limit, or on the institutes cluster, which is very good for testing, but it becomes much more limited when we want to share the data or the visualizations themselves. So yes. So now, please pardon me for the very long introduction. Now we finally get to the visualization. So there are many options for viewing our spatialomics data. And here I have a small selection. This is not everything that it's out there for sure, because the field is expanding, going, and moving very fast. The first three here, Bully, napari spatial data, and Vitessce, we're going to directly see them in this webinar. And then I also really wanted to add other two very nice tools, also open source, like tissue UMaps and MDV. I don't have slides on them today because of time reason, but I would definitely recommend you to just checking them out. They also have their own example data set that you can play around with and see if this could be the right tools for your project. So this is just, again, a bit of things I'm going to show, and I would like to show a lot of example, but of course, the best thing is really when you can try things out for yourself. So I have set up a little repository which contains all the link, all the code, and everything that I've used to show you today. There's well, also a QR code here, but I mean, don't worry if you don't catch it now. We are going to share the slides and the link again after the uh, webinar. So, okay, now I'm going to move ahead. I wanted to just do a very small introduction of the type of data that I'm going to show in our example. So I'm going to show a data set from Xenium. This is a special transcriptomics data set from the run from Xenium machine. And on the same slide, we also run a spatial proteomics experiment using the comet from Luna4. This is a setup, and this is all shared in the bioimage archive, both data sets, and we also have a paper published on this disruption. And this one is a figure from that paper. I'd like to see, okay, it's very nice to have the figure in print and it shows both the protein and the transcript and everything. But now I think we are really coming to the point with this new technologies, the new where just having a static figure, okay, it's nice, it makes the point, but it doesn't feel enough. One would really like to go a bit beyond sharing these images. Oh, sorry, went in the wrong direction. So now I'll say it's time to actually start and do a few things. And I would like to start with Boli as an example. Boli, or Volume Explorer, is a web viewer for 3D volume data. It comes from the Allen Institute, is easily accessible from this address, and it works not really for spatial transcriptomics data. This is an image viewer, so but it is a sport, and I can show you now really well with 2D and 3D images with multiple channels and time series. So if you're doing, for instance, multiplex immunofluorescence or big volume images, or even from the light sheet microscopy side, so really going into different modalities, this could also be quite a nice source. So now I'm going to maybe go quite quickly through an example that I have here. So here I have the viewer. This is the main page, and then what way it works is that one can provide a URL to load the OMSR or your omitive data, and here again S3 bucket would be the ideal way to share, and to cut on a little bit on the loading time, even if it is quite faster. I have it open already here. This is what it looks like. This is our spatial proteomics experiment. I have all my channels here. Here on top we have the 3D controls and everything. In this case it's all 2D, so it's not going to share very much. But what really nice about this is that, well, we are already viewing with not much effort an image that is on the very large side, and we can get quite advanced rendering settings for all the channels. The thing is, after you view and say, okay, maybe I found very interesting set of cells or setup here. I want to then send this visualization to a collaborator, and then I can go directly on, sorry, it was too bit fast, to the share button, and then you can copy the URL, and then it will bring, it will open exactly the same page with the same viewing settings in the same location. So this is also a really nice way to work together. And now I think we should, this is here to speak about spatial transcriptomics. So I'm going to move back to the slides, and then we can go forward with our next viewer. And then, yep. Okay. Slideshow from Canada. Okay, so now we are back on the slides. We have seen our ball example, and I would like to instead move away from the web side of things and go on the desktop. Like to speak about Napari. So Napari is a fast interactive viewer for multidimensional images, but these are presenting in here is that it's actually not just images, because Napari is all Python based, and it has an amazing ecosystem of plugin, meaning that from Napari one cannot just view many different data types, but can also run parts of analysis. And what I think it's especially nice for the spatial data side of things, perform annotations on the images, and also on the cells and the table. And then these annotation can also then be saved and kept in back to our spatial data object. So now we can move to Napari. Here. So here I have the main window of Napari. The way we generally use it is just choose to open a folder with a plugin, open folder. I select my spatial data folder, click OK, and then it will load the interface of Napari. And this is the standard Napari interface here. And since I have now loaded the spatial data object, now I have also these extra bits, which allows me to choose the coordinate system which I want to explore. And this becomes useful if you have multimodal analysis, like in our case, Xenium plus Comet. But you also see it a lot when working with Visium and Visium HD to move between the different resolution levels that this type of data show when you load them into spatial data. And okay, so here now we can start viewing things. Here I have already opened the object to save us some time. So here is our Xenium image. This is just the DAPI for our two samples. But of course, we want to see a bit more. So we can add on top our labels, our cell segmentation, and of course we can share what we want to color it by. Yeah. Okay, and here I can, so now we show it just directly by cell ID. This is just, well, our segmentation. We can zoom in, check our nuclei, everything, but of course want to make it a bit more interesting. So here I also built a very, very basic annotation. I just decided, well, this is one block on the tissue block on the left is one sample, tissue block on the right is another sample, and I can also show it this way. And then, of course, again, we want to be able to explore something more. That is, could be interesting. So then we can move and go to the gene expression. And now I am picking a gene, so CD4 for our, to select a bit of a nice subset of T cells. And so here we see, can already see, okay, most of the tissue is on the purple violet side, so not expressing, but when we zoom in to the stroma, we can now see, this is actually our CD4 positive cell. And these are way this can all work is because the way the spatial data object is set up, all the gene expression and all the annotation that are stored in the table are linked to the labels to our cell segmentation. So it becomes really easy to create these interactive visualizations. And then, of course, we want to also be able to see our transcripts. So here I have added the points for CD4, just to can do a very simple example. They, as expected, overlap mostly with our CD4 positive cell. A bit of a trick here, especially for Xenium, especially if you go for the biggest panels, like the 5K, the latest version. It's generally useful to when viewing to downsample the transcripts a bit, because even if it is very efficient, getting on the billions of points is a lot, and still even showing 1 million or 100,000 transcripts at a time for visualization purposes is generally enough, unless you are really looking at a really rare transcript where then you want to select either those specifically or work on the whole set of points. And of course, now I can hide the cell labels, so we can also appreciate it with just viewing the NAPI. And the end of this example, I also like to show you, and then a bit, because this is our Xenium, but as I said, in this experiment, we also went ahead and worked a bit on the proteomic side. So here we also have the aligned comet image, and this is again thanks to the spatial data format structure, where I can not only save all the elements from the Xenium and the Comet dataset, but I can also save the transformation that is used to register one image, in this case the Xenium one, on top of the Comet one, and same thing with the transcripts and the cell labels that I showed you before. Those are also aligned, those are also displayed, and the transformation is performed in real time. So I also don't need to keep a copy of the registered image of the transformed data set, and then we can go zoom in and see how good is actually our registration. And here we can see, okay, there is still tiny bit of shift, but it's, we can say here, for instance, it's of pretty good quality. So this is our napari, but because napari we can even explore a bit more. There are options to go for also, it's not, of course, full analysis suite, but one can also do a little bit of quick exploration without the code. Here I need to go back to the cell labels. And here I have the option to select what can what I want to show up on the plot. So here I select. So for the x-axis, I want my cell area. On my y-axis, I want the transcript count, and then I want to color by sample. And we can plot it. Here. Okay, this might take a bit too long to plot. Let's see if it is fast enough. If not, okay, here we are. And maybe I'll try to make this a little bit bigger if I can. Sorry, because sharing the screen is a bit on the small side. But here we have a plot. Oh, okay, it's moving. Where I can see which cells I'm looking at from which sample, and I could even make selection and annotation to then save in the object. Here I am just selecting by cell area and transcript count, but of course if you instead wanted to display the expression of your genes, you could already see this one as a way to perform something that you could call like a manual gating, like people used to do for their flow cytometry analysis, for instance, if as a quick and rough way also to do some manual annotation to explore things further. Again, this is just a very quick example. And again, if you have a question, you can come back to it later, but in the interest of time, I would like to move to our next one. And okay, so we are back on the slide, and we have seen how we could combine Napari with the Napari Spatial Data plugin to explore our dataset from a spatial data object, going really quite in depth with our visualization. So just to sum up a bit, this can also be used to view remote spatial data objects. So in this case, everything was done locally, but you could point napari at a special data object stored in an S3 bucket and read it and work to it in the same way. It's quite user-friendly. Lister allows you to do a set of visualization with which otherwise you would need quite a bit of coding otherwise, especially for data sets that go beyond what's directly the output of the Xenium machine or another machine where you could also use the viewer provided by the vendor. This allows you to go a bit further with a bit more complex data set. In terms of limitation, Napari is able to handle 3D images, but this is not yet working on the special data plugin. And the remote support at the moment is still limited to ZAR version 2. The local one with local data one can work instead with the latest version of ZAR and latest version of special data. And here I have also at the bottom a bit of a collection of tutorials, and especially I would like to point the one about alignment using landmarks, which can show very nicely how napari can be leveraged to annotate both data sets, so say proteomics one and transcriptomic one performed on the same slide, and then how this annotation can then be leveraged for the alignment of the two data sets, all without leaving napari and without leaving the spatial data ecosystem. Okay, here I have a couple more instructions on how to view things, how to repeat what I have done afterwards. And again, this will all be shared together with the link to the repository with all the code and links for this setup. And this is also showing again the main interface, loading the data, etc. But this, well, we went through it live. So we would like instead to add to the last tool that I would like to present today. This last tool is called Vitessce. And here we are moving again into web-based visualization. And this is a tool that supports very nicely spatial transcriptomics data, but also single cell sequencing data, and spatial proteomics and 3D data as well. And now it's been expanding more and more also for working with different formats, including a neuroglancer-based viewer. Another nice thing about Vitessce is that it is available using a lot of different ways, but also like Napari. Napari, I showed you the main standalone desktop, but you could also run Napari from a Jupyter notebook, for instance, and Vitessce has also a lot of possibilities like this. It's available as a web app. This is easiest way to just have a look at the examples, and then also share visualizations from your own data set. But you can then use instead Vitessce for your own analysis, for your own exploration as a widget, both for RStudio and Jupyter Lab. So it's been today been speaking a lot more on the Python side of the ecosystem, but here Vitessce would cover also very nicely the R component, and there is not a perfect yet, but a very good compatibility between the spatial data and undata format with the spatial experiment format that instead is prevalent in the R side of the world, let's say. So, yep. Let's go ahead. So, way Vitessce works is that Vitessce allows you to build very complex dashboards, and you can find a lot of examples here at this link. And but this very highly customizable visualization are dependent on setting up a configuration, which is generally built passed as a JSON file, but of course can be built to the JavaScript, Python, and R APIs. And now the Vitessce developers are working more and more on actually generating this configuration automatically, because it can be quite complex, quite a bit of work so they directly guess a good default from undata, omizar, or even spatial data object. And this can seem quite complex, but now at the end, I'll show you how we can then use it, the Python APIs and a couple of additional packages to really simplify the creation of these visualizations without having to learn and work a lot with this complex configuration syntax. So, well, first example, I'd like to show a bit our Xenium dataset through the automatic setup. This is still something that's very much being developed at the moment by the Vitessce dev. And the way this, so it's getting better and better. And the way this works is that through the URL that you use to point the ZAR objects with the spatial data or the undata, you append a little argument that tells the viewer, okay, this ZAR object is in spatial data format, please load things this way. And I have an example here that we can move to. This is, I think, yep. This is this one. So this is our spatial data object loaded directly from the URL. Here I'm just passing our URL. This is public, so you can also type yourself directly. And we have our Xenium image with the DAPI, our cell layer. We can also play around a bit with the transparency. And here at the bottom, I have a list of genes that then I can use. This is now not showing. This is not yet working in the with the automatic configuration. So this is still not perfect, but this is something that then becomes quite easy to use and to set up for a starting point. So you don't really need to write your full configurations and struts. You can get the set this up already this way and use this as a good starting point. But now I would like to move ahead and show you a bit, okay, how we can build upon this and then how we can then make this even easier. So yeah, I'm going to switch back to the slide, and then because what we saw there on the app was first automatic guess, just passing the URL. But when we pass a configuration file, then we can really see much better results. And there is also a page that we can open here that is also contains all the example. Actually, I think this is really nice, and I can show you directly. So there are a lot of ready-made example visualization with everything from single cell data to spatial transcriptomics to the proteomics side to 3D imaging and 3D viewing, and you can select any of them, open it, and you can see the visualization loading, and then you can also go on okay, maybe doing this with Zoom open is a bit much for my connection, but you can go here and you can see this is the configuration file that generated the view before, and then this is already a good starting point to build on. And so let's see if it loads quickly. If not, maybe this is something that you can try yourself directly from there. Ah, no, here it is. And it's quite, I picked at random, but I picked a pretty big one. And this is a quite large single cell sequencing data set. And you can see how interactive this setup and visualization is, and how nicely it is to just being able to share it just as a URL. And here, of course, you can explore interactively the cell sets, so coloring by disease, coloring by gene expression, and this all comes from an undata object. And now I think I'm probably to get towards closing, so I will get back to the slide and go to the last two examples. There are now also two packages. First one is Easy Vitessce. Second one is RPi Vitessce, and this works allow you to generate the configuration file for the viewer and the plots themselves using either the ScamPy or the SpatialData plotting APIs to generate interactive plot, which is generally easier than the Vitessce Python setup, and also much easier compared to writing your own configuration. But here you have to have loaded the SpatialData or undata object yourself. And just to have a quick quick demo. I have my Jupyter notebook here, where to my import of the packages. I hope this is font is big enough. Let's see, can I make this a bit more readable? Yes, sir. Oh, okay, too much. Yes, okay. Okay, so I import the packages, read and load the object. So here is my SpatialData object with the images, the labels, the points, shapes, and tables. I quickly set up the images, and then I call just one function with a few lines where I say, I want this image component with this channel. I want the cell bound to display the cell boundaries on top, colored by sample, and annotation coming from this table. And then this is giving me the setup with the viewer. And then, of course, I can do the same thing, but I can select the gene that I want to display. In this case, I'm always picking CD4 for this demonstration, but you can really go and select any gene from the list, and it will refresh and load. So it's instead of replotting the same thing a lot of time over and over, it's quite quick. And this is not only for the visualization, but we also have you can explore things with ScamPy. So here I have my violin plot of gene expression. You can see the 4 here by sample. So I have my what I call sample A and sample B here. Now I'm displaying, and here again, instead of making a lot of PDFs to share around, I can choose the genes and then from the table, and then it will generate the violin plot again. Apparently I picked one with not much of an expression, but maybe let's see if we are a bit more lucky. But a lot of these here at the beginning are quite empty. Ah, okay, no, here we see, you can actually see that it updates live. And yeah, this is the easy Vitessce one. And last, at the end, very recently, there is this RPI Vitessce tool, which is very similar to Easy Vitesse, but it's not from the Vitessce developers. This comes from the developers of the RPI package, which is a very good package for the analysis of spatialomics datasets, both proteomics and transcriptomics. And just very quick sneak peek, shorter time, very simple. Again, and we use one function to load the object, select which layers I want to show, and then I have a link to click, and it generates already a visualization for me. And this is, again, what we were seeing before, but a bit more complex. Now I've added a heat map, and again, it's all interactive. Oh, I think I like to add and conclude. I think this we will cover. This is very easy to create shareable interactive visualization that highly customizable, and you can reuse the configuration if your data sets are very similar. Main limitation is that this is mostly, especially for the last two examples I show, only with ZAR-V2, and the points are not very well supported. At least there is quite a bit of extra work, but this is going to get much better. This is being actively done. and finally like to say, okay, this is also all coming to public repositories. Here I have an example from the bioimage archive that still in beta, but they are starting to having a spatial-omics gallery with viewable datasets. And this one here is actually the one that we have been exploring together today. And it's also available all from datasets on the UpMap data portal. And I'm not going to spend time on this, but you can register to this webinar, one of the upcoming webinar in the same series, where you can then really see how the UpMap data portal, they manage their own big sets of spatial data. So conclusion, we have a lot of options for viewing, not all compatible with the latest version of SAR, but this is coming. We need to make sure that the transformation are compatible. We need to save the transformation, the transformed image. This works very well in napari, not really yet with on the Vitessce side, but again, will come soon. And now it's becoming much and much easier to share, view, analyze, and collaborate with spatial-omics data in a fair manner. And I would then maybe close here and thank everyone. And maybe we still have short time for questions.
Thank you so much, Michele, for your presentation and for all the demos as well. So we are around the end of time for the webinar, but if I can ask you if it's okay if you can stay a few minutes more and we can take some questions that we have in the Q&A, or if you have another commitment.
No, all good, and sorry for going a bit long.
No problem, no problem. It was very interesting, and I think it was important to have both slides and a demo to understand better how the software was working. So most of the questions were for Napari, but there were also some general ones. There is one participant that is asking if there is a particular viewer that you are recommending if I'm using high performance computing, like Classroom.
So high performance computing, I would say there so you well it depends a lot on your need and the local infrastructure a bit. If you can well, easiest thing could be you could run napari from the cluster and then use SSH with transferring the X session. That way you could get napari running on a cluster node, and if it is allowed, you can get the viewing. Or if you have a Jupyter hub setup available on your HPC node, then you could also either have viewing there with the Vitessce packages, especially so Vitessce or Easy Vitessce.
Thank you. I would take actually another question. I think it's partially connected to your answer already, because another attendee is saying, if I run Napari on a compute server and want to visualize the results on my laptop, how can I do this effectively? I tried use port forwarding, but as expected, it was quite slow. Is there any solution you recommend?
So there, well, it's very hard to discuss without knowing the specific of the cluster and the network. So one could be point forwarding, one would be sharing the X session all together, or last option, but this could also depends a lot on what's available. Might be a full virtual desktop. I don't have a link attender, but you could use the band or bard resources as a publicly available virtual desktop with a lot of image analysis tool, including Napari already pre-installed.
Thank you. The next one received quite a lot of votes is about Volley. So saying, is it safe to upload unpublished data there? So are they stored on a secure server or can I use the software locally?
So they are not storing the data set there. Way it works is you host the data yourself and you then give a URL to the Bully applications, and there the data is read from your own storage. The thing is, of course, it means that it has to be publicly readable. So unless you are going to host the Volley app yourself, it's all open source, it's all on GitHub with instruction. Unless you are self-hosting the app, then I would not use it with data that you are not willing to publish yet.
Thank you. The next one, I think we can take this one. So I believe it's an R user who's asking this question because they say, do you know any packages that can convert spatial objects from R, so spatial experiment framework, to spatial data? So the Python.
Okay, I have to admit, not on the not on the top of my head. Uh, yeah, sorry. Memory is failing me able to do a bit of quick a quick googling. Moment. So
If you'd like to
so
check this later also, I can.
yes, this there are options. Sorry, the cell converted R package from Bioconductor is a cell converter, but with a Z instead of a C. So cell converter,
Okay,
but written with a Z.
for the converter part.
For the converter part between
Okay.
undata and spatial experiment.
Okay. Thank you. Okay, the next one is interesting actually. How would you downsample points for napari visualization?
Okay, then I can. So I take. I hope it's larger enough to see. Maybe make it a bit bigger. Controller here from the console before loading the object. I import napari spatial data and then set this constant here from the console. Maybe, I can make it a bit bigger. This I have the code snippets in the repository from the workshop. So this is the way it's done, but if you want to reuse this, then the code is should be there. Once we share the slides, I'll also send the link.
Perfect. Thank you. I think maybe we can take a last question. We're almost 10 minutes past time. Let me see. So, okay, I think there are two questions that are connected, so maybe we can take them together. So one attendee is asking, does Napari work with direct instrument output, or do you need to shape the data somehow before they can be imported there? And another person that is asking, can I use Napari for visualize H5AD spatial data? Basically, at which moment of my workflow processing can I import the data in napari?
So let's have the raw data directly. No, but or at least not just dragging and dropping, let's say, the folder in with the raw data in napari, but there is the spatial data IO packet, so spatial data underscore IO as readers for let's say most technologies, and those would take directly the raw folder from the instrument as input, and then the object coming from the reader can then be directly opened in napari. And same thing from the H5 AD side. You once you read it, uh, uh, once you load the H5AD object into Python, then you can call napari and it would open and be viewable. There, the question becomes one a bit more of performance in the reading with the ZAR compared to the reading with the H5AD, but that depends a bit more on the specific use case.